as you mentioned, it is a very, very good idea to have one conference in person sponsored by JCMT. So
Yes.
we can gather together. We can start the first one in China, or then move it to Europe, where you have an associate editor who could take care of the logistics. So we can make it in Italy, for instance, and I can help, of course.
Oh, great. We are expected to that. And we value your contribution. Please forgive this. And
don't worry. That was just mentioning that
because the slides is has done big.
I cannot hear you now.
Okay.
Okay, now yes. So I have to introduce Giulia Monticone and...
Hi there. It's Justin
Hi
here.
there.
Sorry. I've been in the
Hello.
waiting room for the last 10 minutes and no one thought to let me in.
Okay, good.
But now I'm in. Really good to meet you. Shall we start? Frida?
Yeah, yes.
Okay.
And
Great.
at the beginning, I will say, dear participants and ladies and gentlemen, welcome to this webinar, How to Live a Better Life of Long-Term Cancer Survivors. And this is Frida, the editor of the Journal of Metastasis and Treatment. It's a great honor and pleasure for me to express our sincere appreciation to all of you for being here. On behalf of our staff and the editorial board, in particularly, we would like to express great gratitude to the webinar chairs, Professor Stebbing and Professor Ciro Isidoro. Thank you for helping organize these big events, and to our guest speakers, strong support. We hope everyone will enjoy this academic webinar. Now, let's warmly welcome Professor Stebbing to deliver a welcome speech. Welcome.
On behalf of Journal of Cancer Treatment and Metastasis and the other speakers, it's a great pleasure to be here today. Really to talk about what we are all keen on, which is how to live a better life for long-term cancer survivors. As we know, although the incidence of cancer is increasing, its prevalence, the number of people living with the disease, is increasing as people live for longer and longer with their malignancies. We're all interested in making sick people get better, improving quality of life and not just quantity. So it's great pleasure that we're joined by 3 individuals who are going to give short talks. The first, Dr. Kundu, is going to talk about navigating long-term breast cancer survival using cost-effectiveness technology in India, which is very, very broadly applicable for the developing world. Next, Dr. Sergey will talk about bridging the gap between medicine and technological singularity. And that's something I think that we're all thinking about with large language models at the moment and the rise of artificial intelligence. And finally, Dr. Carrillo will talk about the challenges of long-term cancer survivors in low and middle income countries, where access to healthcare is sometimes much harder along with its cost. We're all interested in democratizing healthcare, making it broadly applicable, and we're really pleased you're with us today. So we're going to start with Dr. Kundu's presentation.
Thank you. Thank you. Hulda, can I request you to open my presentation, to share it? Or should I share?
Yes. Now you can share your screen. Professor Kandu, can
Yes.
you share your screen?
Yeah, actually, I gave you my presentation. I sent you today. Can you?
Oh, okay.
Okay. Okay. Just let me just try. Open my presentation.
Okay, sure.
Okay, I'm trying. Just give me 1 second. Can you see my presentation?
Yes,
Okay.
you can continue.
So first, I'd like to thank Dr. Justin Stebbing and Dr. Ciro Isidoro, as well as Frida and entire JCMT team. Am I audible? Frida, I am audible.
Yes, we can hear you perfectly.
I am audible, right?
Yes, we can hear you perfectly.
I am audible. Yeah. Can you hear me?
Yes,
can
we
hear
can
me,
hear
right?
you. Yes, we can hear you.
Can you, Justin, can you unmute?
We can hear you perfectly.
Can you hear me?
Yes. Oh, yes. We can hear you.
You can hear me, right? You can hear me?
Yes, 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 yes.
Okay, fine. So I'll be talking about, I also like to thank Professor Lucio Mele, editor-in-chief, who is my long-term friend, to be the editor-in-chief of this journal, as well as be part of that. So next 15 minutes, I would like to discuss about that, that to have a better life of long-term survival, especially especially that, give me 1 minute. No. Just give me 1 second. This one. Yeah, let's go to slide. Yeah, so sorry for that technical problem. So navigating long-term breast cancer survival using advanced cost-effective technology in India. What we are going in India also, that among all cancer, breast cancer is #1, especially women. And as according to Globocan 2020, that breast cancer accounted for 13.5%, which is 178,361 of all cancer cases, and 10.6% death among all cancer-related death in India. So if you see that our survival rate, especially stage 2 breast cancer, is about 60 to 66%, whereas that the global rate is in developed country is 90 and above. Because of India, we have that overall poor detection system, and when patients detect this breast cancer, they are already in that stage 2. So that's why that government of India making multiple efforts in order to increase the survival rate. Coming to the tumor heterogeneity, you know that understanding that TME, tumor microenvironment, not only the tumor cells, there are many other stromal cells, which include cancer-associated fibroblast, CAF, many different kind of immune cells, tumor-derived endothelial cells. They express varieties of genes and proteins, and in a coordinated manner, they control that progression of the tumor at the primary site, angiogenesis, ECM depositions, as well as metastasis at the distant organ. So, as all of you know, breast cancer, there are 4 major subtypes. First one is luminal A, where HER2 negative and ER and PR positive. Second one, luminal B, where all 3 positive. Third one, HER2 overexpressing, where PR and ER negative. And triple negative, where all 3 negative. This is also called basal-like. In addition, there is another one, inflammatory breast cancer, which is not very common in India, but especially India, the TNBC or basal-like breast cancer is that one of that major type of subtypes of cancer in India. And it starts with a little lump, as you can see here and there, and then tumor grow at the primary site, then first target to the lymph node. Lymph node or lymphatic metastasis in breast cancer is the major issue. Thereafter, it goes to lung, liver, brain, and bone. So we are running one national level mega program on breast cancer, trying to understand that molecular mechanism in hormone refractory breast cancer in combination with 6 major national level research institutes funded by Government of India, where we are establishing the primary culture and cell line from hormone resistance and sensitive breast cancer patient tissue. Second, to develop the PDX model, screening new drugs, testing efficacy of selective known drugs, and identifying therapeutically relevant biomarker in patient-derived xenograft or PDX model. Third one, identification and validation of novel therapeutic target from patients or PDX model or primary culture using phosphoproteomics and genomics-based approach. So for your information, we are the first laboratory in India developed that for breast cancer specific PDX model. As you can see, I am showing the representative of G1 to G5 PDX model, and over that increasing number of that this PDX generation, that the tumor grow faster if you go from G1 to G5. And we have characterized to check that from G1 to G2, G3, G4, G5, they are showing the same pattern when we use that ER-PR specific signatures like ER-alpha, PR-alpha, HER2, as well as EMT specific signatures and marker, along with Ki67 and cytokeratin. So coming to one of the genes and proteins we are working last 2.5 decades, it's ECM associated chemokine-like protein osteopontin, interacts with integrin and CD44 and express very high level most of that cancer tissues and cell lines, including breast cancer tissues. And there are several thousand papers showing that this gene and protein act as an early diagnostic marker in breast, as well as few other selective cancer, and also act as a therapeutic target. It basically interacts with CD44 or alpha V beta 3 integrin, regulates cell-cell communication, cell matrix interaction, angiogenesis, etc. So what we have shown, one of my grad student, that like many growth factor or chemokine, this osteopontin also transdifferentiate from resident fibroblast to myofibroblast, and that we have shown using that breast cancer specific TNBC cell line as well as that human fibroblast BJ cells. When we expose that this chemokine like osteopontin with human breast cancer cells, we find that osteopontin transdifferentiate into cancer-associated fibroblast or CAF, and the expression of CAF-specific signatures and markers and genes like alpha SMA, PDGF receptor beta, FSP, FAP alpha, they express more, as shown by Western confocal and qPCR. Then we knock down osteopontin in parental TNBC breast cancer cells and then expose with a normal fibroblast. So we have a parental TNBC, MDA231, exposed with fibroblast, or osteopontin knockdown exposed with fibroblast. And when we use this in that breast cancer cell line, in human fibroblast cell line or that normal associated fibroblast, in parental breast cancer along with the fibroblast, the expression of CAF-specific signature marker goes up. But when we knock down osteopontin with a lentivirus-based approach, we find that CAF-specific signatures and markers are going down. It suggests that osteopontin indeed plays an important role for differentiation of fibroblast to cancer-associated fibroblast. Then we have shown that transcription factor 2:1 plays an important role along with stroma-derived factor 1. So in parental breast cancer cells in association with fibroblast, the expression of HDF and 2:1 goes up. But when we knock down osteopontin and then associates with fibroblast, then that HDF and 2:1 goes down. Then we made two independent preclinical animal model to support our hypothesis. One carcinogen-induced breast cancer model in osteopontin knockout and wild type. Knockout mice develop reduced tumor growth, and another one co-implanted breast cancer parental cell TNBC specific MDA231 along with fibroblast, or osteopontin knockdown 231 along with the fibroblast independently injected to the non-skid mice. And these parental TNBC cells with fibroblast, they grow tumor much faster, but osteopontin knockdown breast cancer cells with the fibroblast, they have a reduced tumor growth, tumor volume and weight. As you can see here, we have analyzed further because of shortage of time. I'm not showing those data. What is important here, we have analyzed that grade 1, grade 2, grade 3 breast cancer patient, check the expression of osteopontin and alpha SMA, CAF-specific, CAF substrate-specific signature. So the expression of osteopontin and alpha SMA goes up in higher grades of breast cancer. But most importantly, in grade 3 breast cancer, we find that alpha SMA positive CAF, cancer-associated fibroblast, their expression is high, and 2:1 also co-localizing in this area, which suggests that alpha SMA positive CAF plays a very important role in that regulation and in the growth of breast cancer in this patient in response to osteopontin, and 2:1 also plays an important role, which we have further proved by tissue microarray, as well as we have analyzed this TCGA data with the bioprotal, and there is a very nice correlation between osteopontin and all these CAF-specific gene expression. So that suggests that osteopontin, irrespective of interacting with integrin or CD44, it regulates that 2:1 activation through ARC or TIC3 kinase pathway, and ultimately controls alpha SMA, HDF1, etc., and then CAF-dependent breast tumor growth. So here, what we are taking that approach in order to increase that survival of the breast cancer patient. We are moving towards that combination therapy where the targeting CAF in association with the tumor cell by suppressing that osteopontin integrin regulated signaling pathways, developing specific druggable molecule in association with immune therapy, and testing in our patient-derived PDX model, and thereafter we'll be moving to phase 1 and phase 2 trial. This will be combination therapy targeting CAF so that we can increase that survival of this breast cancer patient. Similarly, as you can see, started with a peripheral tissue, fibroadenoma, ductal carcinoma and infiltrating ductal carcinoma, protein expression, along with HIF-1 alpha. This also published in Oncology, and previous paper also published another Oncology. So therefore,
Professor, can you hear me? We
I think you've gone on to
now.
mute. I think if this carries on, we should move to the second speaker because he's muted himself. And can someone message him? I think at this point, I'm going to make an executive decision. I was looking forward to hearing this, but he's muted himself.
Frida will send a message to Dr. Kudo.
Yeah. Can we move to the second speaker? Okay. Well, it's finished now anyway.
Justin?
Yeah,
Perhaps
I think.
Sorry, perhaps you could just thanking him for the presentation.
No, of course.
Move on.
Thank you, Dr. Kundu, for the presentation. We're now going to have Dr. Sergi on bridging the gap between medicine. Can you hear me? Can you hear me?
Yes, we can hear you.
Perfect. We're now going to have Dr. Sergi on bridging the gap between medicine and technological singularity. Thank you. Sorry, I think maybe the next speaker is Dr. Pause on towards smarter and kinder drugs using tumor selective strategies. I apologize.
Good afternoon. Can you hear me?
Perfect.
Okay. I've just entered very late because I've had some Wi-Fi problems. So I'm hoping, fingers crossed, that I'm going to stay on here for the talk. Right. So let me share my slides. Okay, so it should now be on presentation modes. So I listened in a little bit on the talk before unfortunately the Wi-Fi sort of destroyed my connection, and I really enjoyed that talk. So and I could see that there was some background to breast cancer. So I'm gonna skip a little bit of my introduction actually, because I think it's just a duplication. I thought it'd just be useful, just sort of put us on the map here. So we are in Bradford in North England, and we are very close to a national park, which you have got on the left-hand side there, Yorkshire National Park. And our institute is basically a purpose-built institute where we have everything from the design of new molecules to take new therapeutics up to a potentially phase 1 evaluation. So I was going to give you a little bit of introduction on the breast cancer, but I think you've heard a very nice background there from the previous speaker there. So I don't need to speak too much about it. But when we are talking about developing kinder treatments, smarter treatments. It's also about selecting the right population. So this is kind of a very simplistic view of the previous talk, but one that I normally give as a slide to people who are not so familiar with different subtypes. But of course, when we are developing smarter drugs, we need to try and target them to the different subpopulation of patients. So in this case here is breast cancer. And what we're particularly interested in our institute is looking at two, I would say, broadly type of therapies. One is looking at potentially proteins or enzymes that are highly expressed in the tumor microenvironment. And another one is looking at yet unproven therapies that are focused on basically providing a non-toxic therapy to stop the metastatic character of tumors at the time of surgery, where potentially cells can obviously survive and escape that. So if we're looking at, for example, systemic therapy, when in the case of breast cancer, you have got the clinical approved drugs, you need to then start looking at how you can target these more aggressive stages of breast cancer. And chemotherapy is still very much used in that space. I've put some examples of drugs there just below the red arrow on some of the drugs that are being used. We have also got targeted drug therapy. So these can include antibodies or antibody drug conjugates. And in principle, these are more selective for the tumor and therefore should also result in lesser toxicity. As with many drugs, even if they're designed for this purpose, there's still side effects associated with these. So we think that we can properly try and mask some of the drugs that we use and try and release these compounds selectively in the tumor microenvironment. And we work with clinicians to try and address this, of course. And I think sometimes it's a bit like a chess game in terms of what you do to the tumor. You know that if you give the patients, hormone targeted drug like tamoxifen, what's going to happen next? If they relapse, then you have your next sort of generation of compounds that might be a little bit harsher, like the chemotherapeutic agents that will then destroy the dividing cells, etc. But can we do a little bit better? Can we help the clinician a little bit better by providing better therapies? And on the right-hand side there, you have the Trojan horse concept there, where you are basically transporting the soldiers into the heart of the battle. And in the context of cancer treatment, it's about, can we actually masking the cytotoxic therapeutics until they reach the tumor sites where they get activated. So again, the previous speaker spoke about the tumor microenvironment, and of course it's extremely complex. I'll try and give you two very brief scenarios of how we could properly exploit the tumor microenvironment. We know that when we treat the local tumor, we will probably be able to reduce the tumor size. Can we completely eradicate the tumor? In some cases, yes. In most cases, patients might relapse. And then when the tumor repopulates and potentially metastasize, you have a much more aggressive tumor. So potentially smarter drugs could be included at the neoadjuvant stage or at the adjuvant stage to replace those drugs that are currently being used in the clinic. So we are particularly interested in some enzymes, and in normal cells, these enzymes are our friends. They do what we want them to do. But the enzyme expression in cancer microenvironment sometimes are hijacking these enzymes so they become our enemies, and that's not what we want to do. So we are interested in the enzyme expression in different cell types within the microenvironment. So MDR, multi-drug resistant cells, cancer stem cells or tumor initiating cells, hypoxic cells, and the proliferating cells are probably those that we can target already. So the first little snapshot is on treating hypoxic tumors. So we don't actually have any clinically used drugs to treat hypoxia. And there's been 8 prodrugs that have been progressed there, 1 from our institutes. And I'm going to show you just the concept of that. But the general problem, obviously, is when you've got hypoxic cells, these are furthest away from the blood capillaries. And let me just see, can I get a pointer option here? Okay, so you have got these cells here in blue. These are round about 100 micrometers or a bit further away, and these are already starting to have very low oxygen tension. They're very much like quiescence. They don't proliferate very well. They survive the treatment because we can't deliver enough concentration to these cells. Similarly, radiation therapy is not particularly effective in those cells because of the low oxygen concentration and therefore generation of radicals that can then subsequently damage the DNA. Okay, so in our institute, we have been working on a prodrug that's gone into a phase 1, phase 2 clinical trial, but has not been progressed further because, unfortunately, because of intellectual property, patent has expired and is on a prodrug called AQ4N. You have it as a structure there on the top part of this, the slide here, and because of this negative charge here, it doesn't bind very well to DNA, but it's very similar to another drug called mitoxantrone, which is also used to some extent in aggressive stages of breast cancer in parts of a cocktail chemotherapy, if you like. So when you have this N oxide, it's negatively charged, and as it tries to bind into the DNA, it can't because it's repulsive to the DNA phosphate backbone. If you're looking at the same compounds, but without the N oxide, the charge at physiological pH is slightly positive. And when you're tracking these compounds in cell, you can see on the top image on the right-hand corner there, the fluorescence is out in the cytoplasm. The nucleus here is on not affected by the fluorescence, and the fluorescence is the prodrug. But when you then have the N or the oxygen on the tertiary amine there removed, the compound now binds very potently to DNA and inhibits topoisomerase 2. And we know that under hypoxia, we can convert that N oxide specifically to the active metabolite AQ4. So you have a selective mechanism of action there. And going back to the clinical trial, there's something that was done back in 2007, 2008. And the prodrug does exactly what you want it to do. So primarily, it hits the tumor, the hypoxic cells of the tumor, and can eradicate those cells and leaves normal tissues unharmed. Now, because this patent expired, we've been looking at some second-generation compounds, and we've developed them, and we're now progressing them, and they're doing also exactly what we want. So we have potentially metabolites like AQ4. These are covalently binding to DNA and damaging the DNA, so the cell cannot be, so the DNA can not be repaired, and the cell will die. We have equivalents that are prodrugs, and these are entering the hypoxic cores of spheroids. So on the left-hand side here, what you see in green cells are generally what chemotherapeutic agents are doing. They bind only in those outer layers of the spheroids and will kill those cells, but all the blue cells that tend to be hypoxic are left untreated. And our prodrugs penetrate straight into the hypoxic core, where they will be converted to a DNA metabolite that can then generate single-strand DNA breaks and double-strand breaks, as you can see in this COMET assay there. And then we've been working with Professor Stuart Martin at the University of Nottingham, and he's been looking at combining our prodrugs. So in the first instance, the prodrug we call KP167, and it does, again, exactly what we've designed it to do. And he's shown, in combination with radiation therapy, that actually this agent is an extremely powerful radiosensitizer. We published it recently in this paper below, and we are in the process of submitting our next paper. And there's something quite unique going on in that KP167 is a much better radiosensitizer than AQ4N, which was tested on patients. And we think that the underlying reason might have something to do with specific DNA damage response pathway defects that are present in some 2D and 3D breast cancer cell line models. Second part project is also using the enzymes, but no longer on hypoxic conditions. We are trying to utilize the same enzymes, but using a biooxidative mode of action. So we're using a sort of a compound or class of enzymes, a class of compounds called duocarmycins. And these you probably will be familiar with because they are very important class of agents that are now being attached to antibodies as a part of an antibody drug conjugate strategy. So we are doing both a prodrug strategy, and we're also doing an ADC strategy with these duocarmycins. So the duocarmycins, they bind in the minor groove of the DNA where they covalently will bind to an adenine. This hydroxyl group there, I'm pointing out now, is extremely important for the activation of these compounds. If you remove this compound, it should not be able to generate the metabolite up here that can bind in the minor groove. So our hypothesis was, okay, if you remove that hydroxyl group in that pharmacophore, you should inactivate it. I'm not showing the data because we have published on some of this already, but by removing that hydroxyl group in this position there, it is no longer able to generate the metabolite that can damage the DNA. And there is 1,000-fold or more difference between this compound and the one on the left-hand side. So we're really deactivating, non-toxifying this type of agent. And then the question then becomes, can we use a P450 enzyme that naturally wants to hydroxylate a lipophilic compound or substrate and thereby activating it? And so, cutting a long story short here, just for the purpose of showing you the strategy, we have a lead compound over here. The hydroxyl group is missing here. And in the presence of tumors that are expressing CYP1A1, which is preferentially expressed in some tumor types, including breast cancer, we can hydroxylate it in the key position here, activate the compound, generate the DNA metabolite that will then create selective kill. What you're seeing here in the middle is basically an autoradiograph of a plasmid that we have radiolabeled to show that the compounds are active covalently binding sequences of adenine as shown here. This is one sequence. So the prodrug itself at up to 100 micromolar dose, which is extremely high for duocarmycin, there's no DNA damage. The activated compound, so a synthetic analog, is able to generate DNA damage down to about 100 picomolar. And if we take the prodrug and we incubate it with CYP1A1 and we extract the metabolite out, we get the exact same sequence binding scenario as with the activated compound. So that suggests that we are generating this, and we've proven this also using LCMS. So that what is really interesting about these compounds here is that they evade most known resistant mechanisms. So if you can deliver them selectively to the tumor tissue, they're really effective at destroying those cells. And for example, here we've got a small panel of ovarian cancer cell lines, one that is resistant to doxorubicin, one that is resistant to cisplatin, and they remain extremely potent. So these are picomolar IC50 values in cells. So they are, even though slightly affected by maybe some potential resistant mechanism in these cells, they remain picomolar active. But the in vivo study I just show you now is using CHO wild type and one that is transfected with the enzyme that can activate it. And I'm showing you the data set here. So we know we can get the activation by 1A1, and now I'm showing you in vivo coming here. This is a single dose just as a proof of concept. So the top graph there is untreated tumors. And if you treat it with a single dose of either the prodrug or the activated metabolite, we get actually very similar tumor growth delays. Where the big difference is when we're looking at the body weight, so as a form of looking at toxicity, In red here, you've got the untreated tumors. In orange up here, you have those that are treated with our compound, our prodrug, and those that are treated with the cytotoxic analog. And the one that is treated with the cytotoxic analog are clearly toxic to the mice. And 4 of these have been tested on patients as cytotoxic agents themselves, and they failed in clinical trial because they were too toxic. So the therapeutic index was too narrow. So we are showing that actually we can deliver this selectively to the tumors using our strategy here. And the same tumors, so the CHO cell line grown up as a tumor here, not expressing 1A1, when we treat it with our prodrug, we have no tumor growth delay. So that shows you the importance of having the expressing enzyme in that tumor. Okay, so we can safely dose with our prodrug, and we are now progressing this in these 4 cancer types here. So in addition to breast cancer, we are also shown in vivo we can treat tumors that are expressing the enzyme in bladder cancer, colon cancer, and head and neck cancer. Okay, so this was one example. We're also looking at another enzyme called CYP2W1, which is highly expressed in metastatic colorectal cancer cells. And we have shown this in vitro and in vivo, but I'm not showing you the data here. There's some more publications coming on that. Just remains me to say thank you to all my the people in my team, national and international collaborators, and to the funding bodies that have supported this work. And thanks to all you listening here, and I'm very happy to take some questions.
That was a really, really interesting talk. Very specific. I really, really loved listening to it. I thought it was great. Just for the purposes of time, we're going to crack on. Dr. Regina Barragan-Carrillo, sorry about my pronunciation, is going to talk to us now about the challenges of long-term cancer survivors in low and middle income countries. Thank you.
Hi, everyone. Thanks for that very kind introduction, Professor Stebbing. So my name is Regina Barragan. I am currently a medical oncologist, and I'm based in Mexico City. So as many of our participants might be wondering, the context in which I do my practice could consider it be very variable in comparison, for example, to what's seen in the UK or what's seen in India. So today's actually my goal is to share like a tiny scope of what I believe would be the main challenges some of our long-term survivors are currently facing and are going to be facing in the not so far future, and as well a couple of ideas on how to address this. Sorry about that. So the term LMICs, as many of you must be aware about, is a term that actually comes from the World Bank data that it's based on GDP. So in this map, to summarize it, everything that it's not like a dark green colored country is considered a low and middle income country. So overall, the total population living in LMICs comes around 3.4 billion people. So as I was, making this talk, I was in clinic, so I had something that made me realize that made me come to a very important realization. So I had two very similar patients. On one hand, I have this 39-year-old woman married. She has 1 kid. She's a college professor. She owns 2 cars. She has a chauffeur. She has a very nice private insurance. And afterwards, I saw this other patient. She was a 41-year-old woman, single with 4 kids. She is currently part of a cleaning staff in a private home, and she moves around usually walking or by bus. And she actually got her treatment based on donations by different, NGOs and very kind people that shared some of their wealth with her, so she could go and get actually the best treatment available that there's currently for triple-negative breast cancer. Both, as you can see, have like a locally advanced disease. Nonetheless, even though both patients were diagnosed with similar diseases at similar stages and received very similar treatment, I came across some important things. So I invite the audience to give some of their thoughts to the following questions, like who of these 2 patients is more likely to develop lymphedema or chronic pain? Who is more likely to develop a cardiovascular morbidity and also metabolic disorders? who is more likely to have developed infertility, and also who is more likely to develop some type of neurological challenges during her long-term survivorship, such as, anxiety and PTSD. And the answer at the end of the day, even though this disease was very similar and the treatment was very similar, is going to be patient 2. And the actual, one of the main motives I've come to that realization is, for example, when you go and see a high-income country, like the classic example, the United States of America, you can see an important proportion of disparities, of socioeconomic disparities. And for example, in the United States, overall as a country, is a high-income country, as you just saw it in the map. 13% of the population live below the poverty line. So even though in similar context, every patient's journey can greatly vary due to socioeconomic factors that are not related to diseases. And why do we have even disparities in cancer care? So actually, it's a very complex problem that could not be addressed only on one end. You have some cultural factors, behavioral factors, environmental, general health. So it's a quite complex problem to tackle. And even more, this problem might be accentuated in the survivorship phase. So usually most of the countries' cancer budget goes either to early detections and mammographies and whatsoever, colonoscopies, and most of the budget actually goes into treatment, because that's our reality. That's how the medical system is currently more based on. Nonetheless, the lesser amount of budget and resources go into survivorship research, survivorship care, advocacy, and clinical practice. So this problem that exists with the budgetary and the disparities in the economic framework is probably even more so during the survivorship phase. So actually, circling back to the title of the presentation, it's more of the challenge of long-term cancer survivors, not limited to LMICs, but overall in resource-limited settings. So some basic concepts of survivorship. Cancer survivor is considered the patient that was diagnosed with cancer from time of diagnosis until the end of life, even though the cause of the end of life might not be related to cancer. This is like the classic description. So around the mid-1980s, Dr. Fitzhugh-Millan was the one that actually made the distinction between 3 important patients. So actually long-term cancer survivors that we have like a time point of 5 years, a very arbitrary time point. If you come and ask me why 5 and not 7 and not 3, that's something that somebody came up and decided that's going to be. But overall, it's a very different phase. And my point in this is that it differs from the other phases, because the acute phase usually is the time around the diagnosis that people are worried about treatment, surgery, radiotherapy. And right now, going to the long-term survivorship, people are going back into their new normalcy. So that's why it's even more challenging, because most of the resources have been already used during previous phases of survivorship. So when we look, and actually echoing a little about the introduction that was very kindly given to us, the number of cancer survivals is going to increase. That's a reality. That's a reality in the States. As you can see, for example, the number of people that are currently alive since the 5 years from diagnosis, 10 and 20 years, which account about 5.4 of the total population in the US. But really, what are the chances of a woman diagnosed with breast cancer to have a 5+ year survival in different countries? So some data, for example, in India and in Nigeria, come to the percentage of around 60 and 40%. So even though this percentage is numerically lower than the United States, overall cancer mortality rates are predicted to decline. So it's long-term cancer survivors are something that every clinician, every oncologist, even every PCP is going to come across. So that's why I want to sensitize everyone about this coming challenge. And overall, it's a very complex system, because you have to deal with financial toxicity, long-term complication, mental health management, quality of life, late toxicities, sorry about that, and also do some health monitoring, not only due to the cancer diagnosis, and finally state a survivorship plan in the best case scenario. What's the problem? So in the Middle East, they did a couple of interviews that actually I think it's going to help me be a little more clear about the problem we're having as low and middle income or regions with scarcity of some resources. We have problems that, first of all, some insurances don't cover all clinics. So it might cover the oncology clinic, but not the, for example, endocrinology, not example, for example, the cardiology or the fertility treatment. So that's one problem. Also, the follow-up times in many countries are not as ideal as they're supposed to be. For example, time interval between visits can differ for standard of care from about 6 months to 1 year and even more. Also, at this phase, probably as people cannot return to normalcy, communication and interplay between other patients might be less available. For example, at the time of treatment and diagnosis, it is very common that they have this, these advocacy groups and even this connection between patients that they see each other during the chemotherapy or during the radiation therapy. But in the longer term, they might lose contact with some of their peers. So that's also another important burden our patients face. And also, the economic situation that is not dependent on them. Overall, like the economic situation on the region of the country might also play an important burden on the patients. And finally, the number of clinics available that are not centralized in many countries, including mine, are a problem for patients. It is not the same to, be a housewife and have the opportunity to have a car and move around than to be somebody that has to take 3, 4, 5 buses to their follow-up visits. And even though if the clinics are not like cohesive, they might have to go to 6 different clinics on 6 different days. And that might mean that they have to take in a short amount of time, a total of, 30 plus buses. So definitely there are some areas that definitely patients that stem from resource limited setting do suffer more. And overall, the cancer care continuum, shows a lot of disparities, for example, in the screening rates, the time to diagnosis, the lack of research and coordinated efforts, the lack of access to novel medication, and a lower rate of adequate timing and surgery. So I want to take a second just to be more clear on the last point. And why is it important? So when you don't have for timely access, let's stay in the line of breast cancer, when you don't have timely access to radiation therapy, what do the guidelines say? What do we do as physicians? So if a patient cannot access to radiation therapy, well, they should receive a mastectomy. Just to say a very brief example. And it is proven that patients who undergo a mastectomy are going to need more frequently rehabilitation services. Another example is, for example, in male patients with testicular cancer that are, stage 1 seminomas that might undergo only surveillance. But if my patient has to travel 3 or 4 buses to get a CT scan every 3 or 4 months, it is much more feasible for them to get chemotherapy, so they avoid many times these very complicated follow-up schedules. So at the end of the day, treatment does impact on the quality of life in the late survivorship phase. And a very astonishing number is that there is also a systematic problem in the healthcare. So they did this study between African-American patients and non-Hispanic whites that overall about 20% less patients that come from African-American ancestry were informed the need for follow-up care after finishing their treatment. So that's a very astonishing number that really reflects the complexity also that comes and the problems that come from the healthcare providers such as myself. So overall, this all joins into particular barriers for survivorship. We're not talking only about the already complex survivorship cancer care continuum, but also we have to take into account these specific details. And is there really an impact on long-term follow-up you might think, well, if they have less access to treatment and they have less access to surgery and whatsoever, so they might not have really an impact in the 10-year survivorship rates. And that's actually true for us. So when we make an estimation by social deprivation in Latin America, this is a very, very nice paper done by a brilliant colleague from Peru. So most research comes either from Brazil, about 70%, and Mexico, around 15%. And most of the research in the long-term survivorships is related either to surveillance of management of psychological effects and management of physical effects. So when looking at the overall quality of the publications, there's still a gap on the development of, for example, on guidelines and things that you can actually apply to your own context. And when we look at the types of malignancies that are present in these papers, for example, comparing to the rest of the world, we have a problem that our epidemiology, at least what's reflected in these patients, does not look like the epidemiology in the rest, at least in the United States of America, where the 3 most commonly associated malignancies with long-term survivorship are breast, colorectal, and prostate. And here we have more of a problem with cervical cancer, leukemia, and thyroid cancer. And are patients even willing to take part of a survivorship plan? Definitely yes. When surveyed, up to 97% of patients are interested in joining a survivorship program if invited. And when you look actually at the NCC guidelines, you can see that, for example, this is a very nice one because they give you example of physical activity and they do all these nice things like pickleball and vinyasa yoga and tai chi. But at the end of the day, they're based on American culture and lifestyle. So this might be a problem when we're trying to extrapolate evidence to some of our particular patients. So what are some solutions that might help aid these long-term outcomes in underserved populations? Some things like bilingual services, caregiver engagement, and cultural preferences have shown to be positive in the engagement of patients in long-term survivorship plan. And also, I think it's important to adapt to oneself's own setting. For example, instead of, for example, recommending pickleball to a patient, I would say rather go play soccer or do a Zumba lesson because of the culture we have here in Mexico. And instead of vinyasa yoga, I would recommend them going around the block with your neighbor, some kind of things that we know that are actually realistic to our setting. Also, it is important development of language for non-English speakers. So there's still a gap. Of course, there are some guidelines made in Spanish. Spanish at the end of the day is the 4th most spoken language in the world, but still there's a gap in native tongues. For example, we don't have any, or we have a very limited amount of tools in Tarahumara and Guava and Nahuatl that are some of Mexico's native tongues. So there's still an important area of interest, and we should seek to training community leaders, introduction of navigation programs, and trust in the communities through the engagement of cultural humility. So cultural humility is the ongoing process of self-exploration with a willingness to learn from others. So I'm going to go into much detail here, but this is one of the methods that has proven to be better in the development of trust between communities, and one as a healthcare provider should be practicing with our patient population, and also the creation of registries. So there's some individual efforts, at least as you can see in Mexico and Brazil, some survivorship care plans in Africa and Colombia. Nonetheless, they are individual efforts. So in the future, we're hoping to make these efforts more of a national perspective, more of a national thing, rather than some individual data for single center registry or 2-center registries. And this is a very nice example of what they're doing in Brazil. So they have these semi-structured script interviews, so that allow the patients to start answering before their consultation. And I think it's very important also to try to look for the development of resource stratified guidelines. As I mentioned, most of the research done in Latin America are limited to what's shown in your screen, mainly related to surveillance and management of physical and psychological effects, but no specific guidelines on only one SP was developed in Latin America. So what I think would be like the ideal future is we start developing according to income setting for this is just a very brief example on how to give a follow-up regarding the clinical assessment of cardiovascular disease in the low setting, and for example in the middle income setting. As you can see, it needs more resources because more resources are available. So I think that might be also a way to address these problems. And at the end of the day, we're looking at changing policies. So for example, there's already some strategic lines in the 10-year plan for cancer control in Colombia that they're looking for improvement in quality of life of cancer patients and also survivors. I'm going to close my thought with the following needs I think are needed to develop in the future, and also like a scope to what I consider would be ideal. So as a healthcare professional, you need to practice empathy and cultural humility with all patients, so you can actually adapt what's already available as evidence and make it more personalized for your patients. We also should seek for increasing research center in minority and long-term survivals, build community-based survivorship programs, start registries to better understand our patient current situation and needs, and finally to create some resource stratified guidelines according to the social cultural backgrounds and the resources available in that particular setting. And with that, I finish my thought, and once again I want to thank the JCMT for this kind invitation.
Hello, I think I can start, right? Or you have to introduce me. So I am the co-chair. Dr. Stebbing had to leave, unfortunately, so I will take over now for chairing this session and start the discussion. I wish to thank first the three speakers. We had three nice examples of how to address these problems. I may start from the last one. I like the fact that it's been pointed out the importance of having a picture of what is going on and paying attention also on the social cultural situations of our patients and having some guidelines helping the healthcare workers to take care of these patients. But we had also two other beautiful talks about new drugs to prolong the survival of our patients. So now I would like to open the discussion, and if any of the attendees have a question for our speaker, please go ahead. So Frida, maybe you can help let me know if there is any of the participants willing to pose a question to our speakers. Okay, so no question means that the lectures were clear, but still, I would like to invite our first speaker because we lost the conclusion. Unfortunately, his microphone was muted. So perhaps Dr. Gopal Kundu could make some comments and on these lectures, and perhaps can also add, if he wish, something on what he was saying, concluding a remark on his lecture, and then we move.
So thank you. So especially in India, that breast cancer is one of the major concerns. And so recently the government of India, through Department of Health and Indian Council of Medical Research, they are coming with a mega program, how to increase and enhance the survival, especially India-centric breast cancer patient. More specifically, stage 2 breast cancer patient, how to enhance their survival from 60% to 80–90%. So our major concern that we are now, whatever database we have across the country, northern part, southern part, east and west, we are trying to have a uniform healthcare system. We are also going to use artificial intelligence, AI, ML, and DL, establishing some algorithm, and to have a national level database for the management of breast cancer patient. And that is the part of the program where I am also part of this collaborative program, where we are going to use the multiple approach. First of all, we like to see that, because here one of the issues, when we detect breast cancer, they are already in the stage 2. So we have to have a good strategic plan, how to detect breast cancer at stage 1 or prior to that. So if we can detect at the early stage, then either we can, through that neoadjuvant therapy or through that other therapy or surgery, we can control that if there is stage 2 or beyond. So therefore, that our early stage of detection, and through this program, we are coming with a non-invasive detection system. And that also we have established by taking that serum from the patient or some biopsy sample, and we are making some PCR-based approach by analyzing that panel of genes with their specific variants, so that we can detect them at the early stage. Similarly, for treatment point of view also, we are coming with a latest type of treatment, especially combination therapy along with immune therapy, in order to control this. Yeah, go ahead, over to you.
Okay, thanks very much for this comment, because what you were pointing out is also somehow connected with what Dr. Regina said about having a better picture of our patients and trying to address their needs and help with, you are saying, also with the help of artificial intelligence and whatever, but the goal is trying to figure out what are the needs around these patients. I would like to call for a brief comments before we go ahead with the second session, our other speaker, because he presented as these kind drugs to help in treating with as adjuvant therapies, which were mentioned now. So if Dr. Klaus Pors can perhaps give a comment on these, just to close this session, please, briefly. Is he here? Oh, yes. Okay, thanks.
I'm here.
Thanks.
Yes.
Thanks, Klaus. I really much, I was intrigued about those drugs. So if you please can tell us how you can envisage their usefulness in the next trials and next therapy to help somehow.
So I think you know, globally, I think we can all agree on that we are edging closer to better treatments. I think I was speaking to some clinicians. I'm actually in Italy at the moment. I was speaking to some clinicians about what sort of treatments we needed for colorectal cancer. And I think where there are lots of things that we can progress on. So in terms of the smarter sort of strategies, I think is, you know, whatever we give to patients, I think this is the general comment. Whatever we give to patients at high doses, we as humans, we don't like, because we're not naturally built for drugs. So whatever we give them, even if it's a very smart drug, it's not gonna be non-toxic. So we have to try and find so we have to try and find a way to manage the side effects as well as we can. But if we can reduce it, so in the UK, we have a breast cancer charity, for example, and their ambitious goal is that by 2050, their hope is that when we treat breast cancer patients, they can live well with the disease. So we're not necessarily talking about, can we really reach cures for all the different molecular subtypes? Could we get to a situation where, for example, you can think of, you know, people with diabetes or something, where you can self-administer the drugs and you can live well with diabetes? Can we get to that in cancer? If we can do that, we are in a very good situation. And I think that is probably where we can work towards. And I think some cancer types are more achievable than others. We can go to pancreatic cancer, where a very difficult task at the moment, even with the smartest type of therapy that you can think of, we are not quite there. So I think we are globally, there are great efforts by the pharmaceutical industry, biotech companies, academic drug discovery centers. I think we are making incremental efforts that are constantly improving on what we have. I think a space where I think there are going to be new opportunities is combining some smaller molecule therapies with immuno-oncology. I think that space we don't understand so well at the moment, because it's relatively new. But I've seen something preclinically where you can start maybe visualizing in the clinic how these can also improve. So I think it's it'll remain a challenge for us to develop these safe technologies so that we can treat, and the patient maybe can treat at home without having to go to the hospital and get IV for hours on end, and where maybe they don't feel as ill after the treatment. I think we're getting there, but we have a long way to go yet. I think it's probably a comment.
Thanks very much for this comment, because I fully agree. I think we need to change the philosophy in treating our patients now, because let me say, I think we've been in the last 15, 80 years treating the disease, not the patient. So we have been focusing on the cancer. We wanted to kill the cancer. Instead, we have to start curing the patient who can survive with the cancer. As you said, maybe it doesn't hold for all types of cancer, of course, and not at all stage. And of course, what was saying our colleague Gopal Kundu, we need to do as early as possible this diagnosis, but then starting to treat the patient and treat the cancer as a chronic disease. This should be the philosophy, I think. So in this, I think putting together the 3 lectures are somehow giving hints to these directions. So I thank you all the speakers. And with this, I think we can move to the second session, unless they want to do a 5 minutes break, but perhaps we can go on because there are just two speakers. So in 30 minutes, we can finish Frida, can you, do you agree if we go on and I introduce directly the second session speakers? Okay, so thanks to you, but please stay with us because I would like to make a general comment at the end. So now it is my pleasure to introduce the two other speakers. In the second session, we have two speakers, and the first one is Dr. Consolato Sergi. He is a pathologist, like me, by the way, and he works at the Children's Hospitals Eastern Ontario in Ottawa, Canada. And then we have Giulia Monticone. She has an Italian name, by the way, and she works at the Department for Genetics in Louisiana, New Orleans. So Dr. Consolato Sergi will give us a talk on this topic, bridging the gap between medicine and technological singularity. Well, that's intriguing, the title. And the second one by Giulia Monticone is Overcoming Tumor-Induced Immunosuppression to Improve Immunotherapy Efficacy. So please, Dr. Consolato, you can start.
Professor Isidoro,
Yes.
Dr. Serge has
Yes.
sent his video to us because he has the
Okay.
urgent surgical
Okay.
today.
So please,
we will
can you-
have to play the video.
The video. Okay,
Yeah.
please. Thank you. Frida, yes, we cannot hear Dr. Sergi. Can you check the audio?
Wait a minute.
And I can write my book on amazing and get started, published by Springer. Later on, the singularity appeared on the cover timeline. Dr. Soledad and I, we started, and other colleagues started in a course on technology and future medicine, and which inspired and attracted a lot of attention and for not only medical faculties, but also medical students and also biomedical students, as well as other students obtaining less for that. I think pathology is key for global health and also for entrepreneurship. It's important to highlight that we have pathology as the key factor to improve health and also to identify food safety and also food insecurity, and drive the new generation population, and also to enhance a better relationship between patient and physician and decrease the stressful burden mental health crisis. NIH and for peace, personalized medicine, participatory medicine, preventative and predictive are key. A few years ago, published together with Professor Mikuc, a few analyses on external quality assurance in pediatric pathology and pathology in general, using different items to identify how key they are. And we identified that the best feedback was specifically using digital pathology for the pathology in general. Then I would like it to just draw your attention how important is the evolution. And obviously, digital pathology is listed for the moment how it's one probably with also AI, the technological singularity in medicine, because of the number of new items that can be discovered in a specific slide, and this exponential curve that I just drew from DNA, cell, human brain, binary divisional computer, quantum computing, technological singularity will be key for the future. Quantum computing and I'm going to be just giving you very, very few concepts, and I just want to give you some information on how this important sector of computing will be key also to make pathology and medicine in general more expedite in the healthy in health, and also to cure patients. Computer technology has evolved over the years. The idea behind the quantum computing is not trivial or deterministic, aims toward the solution of complex problems, and there is a benefit of quantum field, and which I'm going just to highlight in just a few aspects, and it's important to give an aspect about the superposition. The superposition of a single qubit can be forced into superposition with two states denoted for the addition. Usually in a traditional bit, you can have either 0 or 1. In a qubit, you can have both. And obviously, conventional computing has a very limited number of, operations that can be done. Instead, quantum computing has trillions and zillions of operations that can be performed, and this superposition is key for the future of computing and also for the future of digital technology and application of technological singularity in medicine. If we consider, and we started also coding with Python in Quantum Lab, and also our recent publication using AI and in digital pathology and identification of the specific we found out that obviously, if you compare the analog quantum, you have 50 to 100 qubits working on different tasks at the time, and the quantum annealer, your computing system adopting quantum principles, working on one task at a time, and the universal quantum, which is practically as our aim. Our view computing system with 10,000 to 100,000 qubits in conventional way. Quantum computing, despite the benefit that computer offers, commercial or usable computers still need a long way to go. Here are some common problems in the production process. The qubits are still difficult to translate into arbitrary values. Readable qubit for public use, creating a scalable physical system that can support the qubit number. Quantum computing will be key for simulation, cryptography, machine learning, infinitesimal power imaging, life prolongation with microchips, supported transplant, cancer stem cells, and eradication, solution for food securities and food safety, and climate change solution in the conventional early warning system against malaria. 9 steps to patient safety, build a defense culture, support the lead staff and colleagues, integrating risk management protocol in the system, encourage reporting of near misses, involved in communication with patient, involved communication with public, learn safety lessons, implement keys to prevent harm, and control keys to prevent future harm. Controlling as very standardized aspect, we're using plan, budget, CAC for the cycle, but also Biomedical using Six Sigma and this time. The patient safety team should support a risk culture, reporting, inclusion, study, enhancement, and there are different systems that can be integrated in your LIS. And some that have been put delineated in one of my most recent publications using EPIC. Obviously the customer care is key, and obviously there is some aspect in which you want to differentiate between pre and post. That were pre-analytical, analytical, and post-analytical. There are different aspects where actually you can look at the customer care. For this is an example of customer care for formerly fixed protein embedded material, and using in a transplant setting, a cardiac transplant setting, and for rejection, and obviously flow chart key for both lab and pathology. Most medical treatment are designated for the average patient. That's a one size fits all approach. This is successful for some patients, but not for others. Precision medicine, sometimes known as personalized medicine, is an innovative approach to disease prevention and treatment that takes into account the differences in people's genes, environment, and lifestyle. And there are objectives, portability of health data, maintaining privacy, security, and choice, increase access and sharing of health information, support new platforms for internet environment. Key are the application of genomics, pharmacogenomics, and pharmacogenetics. With pharmacogenomics and pharmacogenomics can play an important role in identifying responders and non-responders to medication. This is also crucial for the precision medicine. You don't want to have a drug that practically kills to everybody, but you also wanted to avoid adverse events and optimize drug dosage to that patient. Pharmacogenomics will be the future, specifically for the precision medicine. Drug labeling may contain information in genomic biomarkers and describe drug exposure, clinical response, and variability, and risk for adverse events. Traditional medicine are focused on assumed diagnosis and utilization of medication that have been shown efficacious in the clinical trial environment. Trial and error doses exist in finding the correct medication for the patient. For example, for non-steroidal inflammatory drugs and for the treatment of osteoarthritis. But SNPs are key, the single nucleotide polymorphism, to identify specifically the patient that there will be beta responders or classified as responders for that specific treatment. And personalized medicine mechanics, it's something that has been indicated as linked to the SNPs code, or the single nucleotide polymorphism code, that play a role in our risk of disease. External factors, environment, diet, exercise may also alter this risk, and this need to be known to physicians. DNA variation to determine drug efficiency and efficacy, and DNA variation can lead to differences in pharmacokinetics in the individual patient. The biomarkers and DNA microarray can help identify the approach for the individual. And the DNA microarray technology, the arrange minuscule amount of hundreds of thousands of gene sequences on a single microscope slide. Something product is known as messenger RNA, and DNA microarray technology is the current technology used in as the first approach for precision medicine. What considering a personalized medicine, we need to consider benefit, the action to prevention shift, susceptibility prediction to disease, improvement of disease detection, deter disease progression, customized strategies for disease prevention, increase the efficiency and efficacy of drugs, increase patient adherence to treatment, improve quality scores, and reduce the time cost randomized clinical trials and drug repurposing, and control cost. There are different demands for innovative services in healthcare, and how this will be an increase of technology in for this patient. Reassuring testing, feeling for the patient that the medicine is going to work properly, and also benefit entails also that we'll experience less or no side effects, the quality of life will improve. But limitations are also present. The reimbursement are not fully established, ethical issues concerning confidentiality, privacy, integration of pharmacogenomics with the taxpayer, and inappropriate clinical education on basic sciences, and missing holistic approach with regard to the interaction of multiple genes in response to the drug. Alternative, well, you can use a lockdown switch and don't do anything. But there, fight yourself, taking the less traveled road, step in inspiration on novel. The child, don't be afraid of failure, as long as you learn from it. And be flexible and adapt, but keep immutable second principles, and expose your ideas, and discuss, argue, and promote dialectics all the times. This is one of the books where precision medicine took place for pediatrics, and Nathan Springer, and all royalties go to pediatric family charities. I would like to thank you, all children and family, the Bukri and Frau Derta, my research labs, the Children's Hospital of Eastern Ontario in Ottawa, my mentor, Professor Mikasa of the University of Innsbruck. And thank you for your.
So thanks very much. Dr. Consolato wasn't really here with us, but he sent us this video. And now I would like to introduce the second speaker, and then we will have a short presentation of the journal, and then a closing remarks. So I would like to invite now Dr. Giulia Monticone, please, and she should be ready with her presentation.
Thank you, Dr. Isidoro, for the nice introduction. So hi, everyone. I'm Giulia Monticone. I'm a postdoctoral researcher at LSU Health Sciences Center in New Orleans. And today I would like to talk about my research on overcoming tumor-induced immune suppression to improve immunotherapy efficacy. So as you probably already know, cancer immunotherapy has revolutionized the field of cancer research. And there is currently a major effort in trying to apply immunotherapy to the clinic and for the treatment of cancer patients. However, responses rates to immunotherapy still remains variable and limited to small groups of patients in different and several types of cancers. So how do we improve the efficacy of immunotherapy? One of the major factors that limits the efficacy of immunotherapy is tumor-induced immune suppression, which is prominent in aggressive solid tumors. Immune suppression is the ability of cancer to suppress endogenous anti-cancer immune responses, as well as immune responses elicited by immunotherapy. And tumors achieve immune suppression in different ways. For example, they recruit immunosuppressive cell types, or they overproduce immunosuppressive metabolites, including adenosine, which my group is currently studying. So you can imagine that one way to improve the efficacy of immunotherapy would be to design therapies that are able to overcome tumor-induced immune suppression. So how do we achieve this? Our group and others have shown that adenosine is an immunosuppressive metabolite that is overexpressed in the tumor microenvironment, and it suppresses CD8 T cells' anti-tumor immune responses. Specifically, adenosine activates the adenosine A2A receptor in CD8 T cells in the tumor microenvironment. And our group showed that this leads to downregulation of Notch1, which is an important regulator of T cell function. And this leads to downregulation suppression of effector functions. So we propose that finding strategies that are able to restore Notch1 and so effector functions in CD8 T cells would be a good strategy to promote resistance to immune suppression and more potent anti-tumor immune responses. To achieve this, the first thing that we tried to test was to identify what is the negative regulator that downregulates Notch1 in response to adenosine in CD8 T cells. So by using a proteomics approach and other approaches, we found that the ubiquitin ligase CBL-B is the negative regulator that ubiquitinate and degrades Notch1 and leads to the suppression of effector functions. So we thought that CBL could be a promising target, and we wanted to test whether blocking CBL-B could actually restore Notch1 and so effector functions. So first, we try to test whether CBL-B knockout primary CD8 T cells are actually resistant to tumor-induced immune suppression. So we treated primary knockout T cells with CGS, which is an adenosine A2A receptor agonist to induce immune suppression. And as you can see, in these cells, Notch1 was not downregulated, and also both proliferation and cytokine production was rescued from immune suppression, suggesting that CBL-B knockout T cells are indeed resistant to adenosine-mediated immune suppression. So we were very happy about these results, and we wanted to test whether a pharmacological approach would be suitable to block CBL-B and rescue Notch1. So we started a collaboration with Nimbus Therapeutics, which is a biotech company based in Boston, which produces CBL-B inhibitors. And we tested the inhibitors in primary CD8 T cells And as you can see, the inhibitor was able to rescue Notch1 proliferation and production of cytokines, suggesting that we are able to rescue effector function in CD8 T cells. Importantly, we also found that the inhibitor did not increase Notch1 level in cancer cells, suggesting that this strategy has a good selectivity towards Notch in immune cells. We then wanted to test whether Cbol-B inhibition can translate in more potent anti-tumor T cell responses. So we use tumor-derived organoids, which are novel ex vivo cancer models. And as you can see, we found that Cbol-B inhibition significantly increased cancer cell death in organoids derived from triple-negative breast cancers and colon cancer. We found that this was because there was an increased infiltration in CD8 T cells in the organoids, and these T cells expressed a higher level of Notch1 and so had more potent effector functions. So we published this data last year, and now we are moving forward to test Cbol-B inhibition in preclinical triple-negative breast cancer models. And as you can see, we were very happy to find that Cbol-B inhibition is able to reduce tumor growth and significantly induce necrosis in the tumor and apoptosis of cancer cells. We also try to analyze the tumor microenvironment. And as you can see, the treatment gives rise to an overall rearrangement of the microenvironment. But today, for the sake of time, I will only focus on the T cell component. So as you can see, the treatment was able to increase the number of T cells. And interestingly, we found that the number of exhausted T cells was reduced in the treated group. And we found that T cells in the treated group, they express a lower level of PD-1, which is a known exhaustion marker. We also found that the number of central memory T cells or stem cell memory T cells were increased. And especially this subgroup of T cells is of particular interest because it's associated with increased Notch1, which we actually observed in tumor-infiltrating T cells, and is also associated with more potent anti-tumor immune responses. And not only this, is also associated with long-term T cell and protective immunity, suggesting that we may be stimulating a long-term protective immunity against cancer. And this could be one of the mechanisms elicited by Cbol-B inhibition. So in summary, we found that Cbol-B inhibitors can restore Notch1 and effector function in CD8 T cells in the tumor microenvironment, and this leads to resistance to tumor-induced immune suppression and more potent anti-tumor immune responses. So you may wonder now, how much is this relevant to the clinic? So we found that Cbol-B has a high expression in T cells in cancer patients. And the expression pattern is very similar to the one of PD-1, which is a known negative regulator of T cell function. And it seems that the pattern is also particularly significant in breast cancer, which is in agreement with our preclinical data. We also try to test whether Cbol-B inhibition can be combined with immune checkpoint immunotherapy, such as anti-PD-1, anti-PD-L1, which is already used in the clinic for small groups of patients. And we found that Cbol-B inhibitors are able to enhance the efficacy of immune checkpoint immunotherapy. This is very important because it means that by applying this treatment to the clinic, we may be able to increase the number of patients that can respond to standard immunotherapy. So in conclusion, our findings show that the Cbol-B Notch1 axis blockade is a novel promising form of immunotherapy. And this treatment has the potential to increase response rates to immunotherapy by overcoming tumor-induced immunosuppression. And this could ultimately increase the number of patients that can benefit from immunotherapy. The treatment has also the potential to elicit long-lasting protective immunity. And so this has the potential to ameliorate the life of cancer patients, cancer survival, and also prevent cancer recurrence. I would like to thank my mentor, Lucio Miele, all the members of our team, our collaborators, and the Journal of Cancer Metastasis and Treatment for this invitation. I thank you all for your attention, and I will take any questions. Thank you.
Thanks very much for this clear presentation and the interesting data. And this goes again into the direction that Dr. Klaus Porz was just saying at the end, when he said we should improve somehow also the immune response because we should try to lower the dose of chemical drugs. And indeed, this is a good example. We can try to have, say, a synergism between treatments so that we can lower down the actual concentration of each of them. And with this, we will lower the side effects and improve the efficacy of the treatments. Let me see if there is any question from the attendees, the participants, and so I ask our managing editors, Frida or Victoria, if they can help to catch some questions from the chat for our speakers. And if not, we can go on with some comments. If you agree, I keep the microphone to say that no questions. Okay, but indeed the lectures were pretty clear, and I think, oh, but we have Klaus, Dr. Porz. Please go ahead.
Okay, thank you. Sorry, I was looking for how you actually activate a hand here, and I struggled a little bit. It was sort of hidden at the bottom. I really like that presentation. There were lots of really interesting things to discuss and sort of think about. So obviously, as we discussed a little bit earlier on, there's a lot to learn about immunotherapies, and here you are proving that there's some links that we can exploit. I was interested in this sort of long-term effect, and I know you haven't got the answers to this, and we can speculate on it. So the NOTS pathway is obviously also one that is very interesting, the stem cell, and you refer to these memory stem cells. But NOTS is also, if you inhibit it, you have an impact on, there are different types of cancer stem cells, and it depends on the cancer type. I wondered whether you could speculate a little bit on whether when you try to intervene NOTS1 or NOTS pathway, what are the implications of eradicating what I would call a malignant stem cell, not one that is protective. And it's a very fine-tuned balance between the two, I guess. Is that something you could speculate on?
Yeah, I think so. So indeed, in the lab, we are working on basically both strategies. So we have strategies that inhibit NOTS1 and strategies that try to stimulate NOTS1, like the one that I presented today. And the important thing about NOTS pathway is indeed the selectivity. So in order to have a NOTS-based therapy, we need to ensure that this therapy has a very good selectivity. So for example, today I showed that we tested this drug also on cancer cells, and we don't see modification of NOTS pathway in cancer cells, meaning that this regulatory axis is not present in the cancer cells, but probably more in the immune component, so it has a good selectivity. However, it's also true on the other side that when we use Notch inhibitors, then those are effective in basically reducing the growth of cancer stem cells, for example, in breast cancer. But again, one of the main side effects of these compounds is also that they suppress the immune system. So it's always the case of trying to improve these Notch-based therapeutics so that they can be selective for either the cancer cells when we want to inhibit Notch1 or the immune component when we want to stimulate immunity.
Okay. Yeah, I think that's I think that you're right there. Can I just it's a technical question. You had these patient-derived organoids. Do you use these? I mean, I don't know how you reproduce it, because once you have used them, they're not like immortalized cell lines. You use them once or twice, and then is that it? Yes. So do the data from your different organoids then, are they very similar? Because obviously they come from different patients, and therefore I wondered whether they respond in various ways or they're very consistent.
Yes, so one thing, because we are working on immunotherapy, so of course, depending on, let's say, if a tumor has a lot of immune infiltrations already, probably in the organoids, you're going to have more T cells, and so you are going to have a better response. And instead, the tumors that are more or less, they have less immune components, then we see a lower response. However, this in a patient would not be a problem, because we aim to stimulate the systemic immune response. So in theory, more T cells should be recruited to the tumor microenvironment and be more effective.
Okay, all right. Thank you.
Thank you for your question. Thank you.
Okay, nice discussion. So maybe we are just a little bit beyond the schedule. So if you agree, I would like to make some concluding remarks on this topic, and then I will present the journal. So first, I would like to go back to what was the discussion with Dr. Klaus. When we said we have been focusing much on just the cancer and not so much on the patient. And we were thinking of starting to think of cancer as a chronic disease to be treated in a different way, not so aggressive. And in fact, if you agree, I think we should consider, for instance, to improve the quality of life of our patients, of our survivors, also to consider the other aspect. We didn't discuss this time that cancer cell dormancy and tumor dormancy are two aspects that should be taken into consideration, not just because I work on that field, but because we think that indeed, once you have done a great job in shrinking the mass of the tumor, the primary tumor the surgeon has removed as much as possible, then we can manage the remnant disease, trying to keep it dormant or slowing down the growth, for instance. For this, it's important also to introduce some other supplements instead of drugs, improving the immune system response, or say putting cells in tumor cell in dormancy. And we need also to do some palliative care for our patients and try to treat the side effects and so on. So there is lots to do, much to do, and this goes back again to the talk of Regina about the guidelines that we still need to treat the survivors appropriately. Now, if you agree with me, I hope, and there are no questions, I would move to the last part of this webinar, thanking you all. But first, please, let me introduce you to the journal. And so I kindly ask Frida and or Grace to start with the PowerPoint presentation of the journal.
Okay, thank you, Professor Isidoro. I will share the screen for you.
Thank you. Okay, thanks very much. So you can see this journal has been set just a few years ago. I've been one of the earliest associate editor joining the editorial board on invitation by the editor-in-chief, Lucio Miele. And you can see that as of now, the journal has published so many papers, and the size score increased with time. And indeed, we are expecting to have the impact factor in next month. And we are quite confident that it will be a good impact factor, maybe around 5. So next slide, please. And let's go to the second slide. Yes, this is to show you the members of the editorial board. And you can see that are represented so many countries over all the world. So not only, you can see we have from United States, from Italy and France, Belgium, Korea, Germany, and so on. And you can see also that our editorial board members, they have quite good HI index, meaning they are very well renowned scholars. Please, the next one. Okay, here is just a list of few of the main, few of the members. So we have the honorary editors-in-chief, and then we have associate editors, and then we have editorial board members. I'm not going to name all of them, but I'm here, the second one. Okay, let's go on, please. And we have also a section for young editorial board members, and they are starting to work with us. And so they are also representing different countries, and they are being joining us and being ready to take over with time as senior editors. Let's go on, please. I'm presenting this journal also to convince you that it is worthy to consider JCMT as a journal where to publish your next paper. Please, the next slide. Hello? Can you please go on, or is there a way so that I can move it? Okay. So you see how many good authors we have publishing in this journal. Please go on. And if you look at the readers, this tells you that the journal is quite well received in many countries, more than 200 countries, where we have our readers, and you can see the number of downloads of the papers published in these journals. So Here is brief history of the journal in terms of indexing, starting from the American Chemical Abstract, then Scopus, and now we are indexed in C. clarivate. So we are expecting, as I said, the impact factor next month. Okay, let's go on. Yeah, it's important also to stress that the journal runs many special issues dedicated to a specific topic. Here is one example, and you can see the how successful has been this particular special issue with so many publications, papers, 31 papers, and more than 600 citations. Let's go to the next. Is another example of other cited articles, and you can see that we have articles cited up to 300 times in less than 3-4 years. Okay, we can move on, please. And here is, as I was saying, another example of special issues. And this also has been quite successful because with 8 papers, it got more than 445 citations so far. And again, in this particular special issue, you can see there are papers cited up to 300 times. Okay? This is also to say that the journal is widely spread overall worldwide, and so it can be read by many people. Here are some ongoing special issues, and by the way, these are the ones where I am acting as the guest editor. Please consider it, like this one for artificial intelligence in cancer treatment and prediction. This is dealing with autophagy cancer, so it has to do also with dormancy, as we were saying before. And this here is an example of how the journal can cooperate in organizing a conference or promoting the conference. In this case, the journal has been the media partner of the conference that I have organized here in Italy, in Novara, just yesterday and the day before. And we had 4 years ago a similar collaboration where also we had a special issue and we published some papers that are quite well cited as well. Okay, next slide, please. And these are other examples of ongoing special issues that you can consider for your submissions, depending, of course, on your interest and field of research. There is this on cholangiocarcinoma, for instance, or specifically on cell death and treatment of solid tumors, which is, you can see, run by Dr. Justin Stebbing, our co-chair in this webinar. And you can see here, Consolato Sergi, our previous speaker, is a guest editor of this other special issue on hepatocellular and cholangiocarcinoma. The next. And the journal also sponsors the webinars, and actually they plan to also have conference in person. And the webinar of today, okay, I was chairing together with Justin Stebbing, was started by Justin Stebbing, the webinar we just had it. But you can see this is a webinar from the editor-in-chief, Professor Lucio Miele, on obesity and diabetes in breast cancer, and so on the others. So let's go on, please. So this is the one we had just today, and then we are going to collect papers from the attendees, from the speakers, And I think it's been quite successful on what I could see based on the number of participants and also based on the quality of the lectures. Okay, the next one. So these are some wonderful presentation, and in other webinars, then we can go on, please. And by the way, the microbiome has been neglected today, but again, is part of the strategies with the diet for improving the quality of life of the survivors indeed. And the journal has also posted some interviews with associate editors and the editor-in-chief. You can see here there are some examples of this interview posted on the website and in the other social media. Okay, let's go on. And in fact, I was just saying the journal spends lots of energy to promote the articles through these social media platforms like Twitter, LinkedIn, and others. So this makes the article visible to a large audience, and this will increase the chance to be cited, but essentially to contribute to the knowledge in the field. Let's go to the next. And okay, this is an example of what I was saying, the partnership of the journal with the conferences. And this is the one I was mentioning I had organized in Novara just a few days ago. And the journal set a special issue to collect the articles coming from the lectures. And you can see here other examples. Okay, let's go on. And here to sum up all the advantages of publishing in this journal, you have a fast and rigorous peer review. I can tell you it's quite rigorous. As an associate editor, I do the pre-check and then a double check again of the reviewers' reports to make sure that only high quality articles are accepted for publication. And this is indeed what is bringing up the impact factor. And the journal provides also a discount for good quality papers that from authors that got good citations. And so this is to encourage the submission of other articles from those authors. And as I said, the journal takes care of promoting through the social media platforms the articles. And again, they are also considering to give a best paper award annually, which will be selected by the associate editors based also on suggestions coming from readers and based on the citations, of course. And so the message is join us on this in this journal. Next, and I think we are going to the end, correct? Yes, yes. So you see, this is the last slide, I guess. So we welcome you at the journal to join us submitting your paper as soon as possible. Okay? So with the next slide, I think we have done, if Victoria confirmed, yes, thanks for your support. And so I would like now to thank all the speakers again. They've been with us. They've been participating in the discussion. Thanks very much for interacting with us. And I thank also all the participants, and I leave to Victoria closing the webinar.
Thank you, Professor Isidoro. We appreciate for your help to chairing this great webinar. I hope everyone enjoyed this webinar, and we also appreciate for all your contributions and for the journal, and also our speakers. You are all the editorial board members of our journal, and we do appreciate your support and the contributions in the past years, and we expect to have your continuous strong support in journal's future better development. Thank you so much for your effort for and help for this successful and great webinar. And our journal will organize more webinars in the future, and we hope to invite you to join our future webinars again. Thank you.
Okay, then thanks to all and see you soon. Bye-bye.
Okay, thank you for your great presentations from our speaker. Okay,
bye-bye.
Thank you. Bye. Have a nice day.
Bye-bye, Klaus. Bye.
Bye-bye.
Bye.
Tốt lắm.